Okay, thank you so much for the invitation and introduction. I feel so honored to be here. So I first knew MGH as a file extension of the Brain Service Map. Then when I visited Boston and saw MGH and stopped on the red line, I realized the file extension is named after the hospital. As FMR research, of course, I have to go to visit the shrine where the 3 server was built. Then I took the subway, got off the Charlie's MGH campus, and walked around the main MGH building without realizing that I was in the wrong building. What I should have done is look at the map. Maps are so important for the tourists, for guiding us to where to go, not only for tourists, not only just for tourists, but also for the neuroscientists. Today I'm going to talk about the work I've done mapping the brain regions, brain regions of monkey dedicated to the object recognition. Here is the side view of the macaque brains. Object recognition happens in the ventral visual system. It starts in V1, then goes through the V2, V3, V4, then the inferotemporal cortex. We know that the IT cortex has the high-level object code for high-level code for visual objects. One of the ways we know this is a stroke in this part of IT brain can cause a very selective deficit in the object recognition. For example, destroying a person's ability to recognize faces. Area in the early visual cortex have a retinal-topical organization, which means the neuron that shares a similar reset field location in the regions in the retina are grouped together. This was discovered through a pioneer experiment here at MGH. However, the IT organization of this part of brain is still not clear. In the late 1970s, Charles Gross discovered a single cell in IT cortex that are selective for specific complex form, such as hands, trees, and faces. And here is the famous face cell, face select neuron that Gross recorded from. They respond to human and monkey face, but not to hand or toilet brush. 2 decades later, Nancy Cavendish discovered the face area in the human brain that responds more when subject look at a picture of face than when they look at a picture of other objects. Let me remind you again, this work was also done in MGH. And it turns out that a macaque monkey also have the face patches. If you show the picture of faces and object to a monkey inside the fMRI scanner, 6 patches will light up selective to faces. Shown here in yellow, this work is also done in MGH. Professor Tutel, I think, and Professor Wald are the co-author for these papers. Electrophysiology recording in these patches show that almost all the cells in each patch are face selective. Here the response profile for all from a face patch of 1 monkey. Each row represents 1 cell and each column 1 image. And the first 16 columns are response to faces. Red means a strong response and white means no response. As you can see, almost all the cells are face selective. The 6 face patches are strongly and specifically connected to each other. We know this from the microstimulation experiment inside the fMRI scanner and also from direct tracer injections. The patch perform different steps in face processing. In particular, cells are increasing view invariant as you go anterior. In the middle face patch, single response to face at a specific view. Here is an example neuron that only do response to face when the face is looking at the left. Where in the anterior patch, cells respond to across all the views, as shown for this example neurons. Besides face network, specialized networks have been found in IT for bodies, things, and colored objects. In my talk, I will address 2 questions. First, what's the functional purpose of specialized network? In other words, why does the brain contain the face patch, body patch, and so on? I'm going to present evidence that a specialized network allows the brain to see objects much better in clusters. And the second, what is the overall organization of IT cortex? I'm going to present new findings that IT is actually organized into a systematic map of object space. And those discovered category select regions are just one part of this map. First, let me discuss the functional purpose of these special networks in IT. In general, it has been proposed that the category selective regions exist to minimize the wiring lengths for neurons that need to communicate with each other to represent a face or body. For example, to accomplish lateral inhibition. But this is a fairly fuzzy idea. I would now want to present a very specific hypothesis for why category select regions are hugely beneficial, which is that they allow the brain to break down a cluttered scene into the parallel channels. In real life, it's not very common for you to see only one object appear in the visual field. Most time you will see something like this. It would be much easier if the brain can break this complicated thing down into the parallel channels, so it could just focus on the faces and the bodies and so on. What I will show you is if we have two gradients, category selective region and normalization, that give you a winner-take-all circuit. In other words, we can achieve this goal, breaking the cluttered thing into the parallel channels. Now you know the punchline, let me explain the evidence for this. We recorded from two category selective regions, a face patch and a body patch. As a simplified model of this cluttered thing, we present two objects, a face and a suitcase. We present two stimuli in four spatial locations, so the face could be contralateral, ipsilateral, above or below the fixation point. Here, the contralateral or ipsilateral face refers to the recording site. For example, if we record from the right hemisphere, the contralateral means the stimuli will be presented on the left visual field. And both stimuli were shown at a range of contrasts. Here's what the stimulus look like. The monkeys were asked to keep fixation on the center of the screen to choose reward. Stimuli were presented in a random order, including isolated faces, isolated objects, and compound images at different contrasts. We're measuring the firing rate for each conditions. And here the averaging response for single isolated faces at increasing contrast. And here the average responses for the single isolated object at a decreasing contrast. Now the question is, when we put the face and object together, how will cell respond?
Is there a reason you did a human face instead of a macaque face?
No specific reasons. People found that those cells also respond to human face and macaque face equally well. Yes?
So the statement that there are 6 face-selective areas, do you find that in every monkey image?
No, they do exist some random, has some fluctuation. Some monkey only found 4, 5, 4, 3. Some monkey found, most of monkey found 6 patches.
I see.
Yeah.
Have you done cytoarchitectonics? That is, are these patches cortical areas in the strong sense? Do you find architectonic differences between a face patch and the adjacent house patch or object patch?
You mean the anatomical?
Yeah. Do you find the layers change content or shape?
No, we don't do the. We don't. I don't remember. Yeah, we haven't done some anatomical for those patches. We don't think there's a clearly structural difference between those patches to the adjacent patches. We think that nearby maybe have the similar structure. Its only function is different.
But you see differences in connectivity.
Yeah, we do see the different connectivity. Right. Okay. Maybe if I explain that later in my later model, you can see a full picture about that. Okay. Okay, now the question is when we put a face and object together, how will cell respond it? And here the black line represents the response to face-object pair. For example, the leftmost point represents the response when we put 10% of contrast of face and 90% of object together. As seen in this figure, this black line is very similar to the red lines, which indicate the neuron in the face patch performing a winner-take-all behavior. What would happen if we just switch the face and object positions? Now the face is presented in the ipsilateral visual field. And here is the responses for the faces alone. And here the response where objects are presented in the contralateral visual field. And here the black dots represent the response of the face-object pair. So now you can see the behavior is totally different. Now the response can be described as a weighted averaging model. What happens when the face and object are vertical aligned? No matter where the face at the top and the bottom, cells always perform winner-take-all. Here's a summary. Cells in the face patch always show winner-take-all behavior in every case except when the face are in the ipsilateral visual field. But what happens when we record from a body patch? Now we use a monkey body as a region's preferred stimuli, and the response of body patch are marked as these green dots. And the response of non-preferred image now is the face is marked as red dots. As seen in this figure, we observe the exact same pattern as we observed in the face patch. Only when body are presented in the ipsilateral visual field, we see a weighted averaging behavior. But in most other cases, we saw the neuron performs a winner-take-all behavior. How does the brain achieve winner-take-all behavior? Using face example, and here the response for when the present face alone, and here is the response for object present alone. And as you can see, also neurons show the strong preference to faces. It still shows the response. It still responds to object. When we put the face and object together, how could neurons shut down the input from the object stimuli? People might think neurons need a certain filter or use their attention to do the filter things, but it turns out the solution of the brain is much easier than that. This pattern exactly what would happen if you just combine the category selectivity region with normalization. With these both gradients, we can get a winner-take-all. Some of you may have heard of normalization. It is so-called canonical computation that exists in the neural system across different species. And here the response of the neuron recorded from the fruit flies olfactory system. The neurons are less sensitive when the mask order is stronger. So this can be described by a normalization model. When the mask order gets stronger, the whole turning curve moves to the right, which means less sensitive. And also in monkey V4, when two stimuli are presented in the reset field simultaneously, the response of cell can be described by a normalization model as a weighted averaging model. But when the monkey is starting to pay attention to one of the stimuli, say stimuli 1, now the response will be more like the response of stimuli 1 presented alone. Such phenomenon is called a bias competition model, and it can be understood as attention. Attention changing the gain of stimuli 1 with a normalization model. Returning to IT Cortex, suppose we have a face cell and it is getting input from two stimuli. Both groups provide excitation as well as a normalizing inhibitions. And here is what the normalization equation would look like. C1 represents the contrast of face, C2 represents the contrast of object. And A is a response to face when it presents alone, since if you set it to C2 0 are roughly equal to A. And B is a response for object presented alone. Now we are adding one more parameters here. It's the strength of input from the W as the strength of input from the neighbor neurons. Such weight can be understood as the sum of the response of neighbor neurons' responses. So when the weight of face patch, when the face input is much larger than the weight of the object input, the response will be more like when the face presents alone. That's what we call the winner-take-all model. So when the weight of the face input is similar to the weight of object input, now the response will be more like the weighted averaging model. How can we measure this weight? Since local field potential is generated by the sum of electrical current flowing from nearby neurons. So it should be a good indicator for the weight. So here's a LP amplitude when face and object are presented alone. And we do see the W1 is much larger than W2. So that's why you can see the winner takeoff behavior. Now when the face are in ipsilateral and the weights for the face are in the ipsilateral and the object in the contralateral, their weights are similar. That's why we observe a weighted averaging model. Such difference is because of neurons in IT cortex still reserve the strong contralateral bias. In the vertical configuration, LFP also can explain the observed winner takeoff behavior. So that's why category selectivity plus normalization give you the winner takeoff behavior. Yes.
So typically the winner takeoff require a nonlinearity.
You require, what do you mean by?
Well, I mean, winner takeoff implies.
Yeah, inquires a nonlinearity, right?
So where in your.
No, this equation, if you W1 is much larger than W2, then this normalization, this division part is already nonlinearity. If you have this division part, is also nonlinear. If it's linear, you have two stimuli, the response should be sum of the response. But because of a normalization, that's why you never get a sum response for two stimuli.
It's not quite winner takeoff. So you forget that there's small activations.
No, you have, if you W1 is much larger than W2, sorry, it should be like this one. If the W1 is much larger than W2, then basically you can surround the W2 part. That itself give you a winner takeoff. Yeah. The normalization model itself is not a linear model, so. Where are we now? Okay. So you might wonder why face cell did not perform winner takeoff for the whole time. For when faces are body in the ipsilateral visual field, we saw weighted averaging behavior. But remember, brain has two hemispheres, and in the opposite hemisphere, the cell will be doing winner takeoff. So brain can easily rely on the cells from the contralateral hemispheres. To summarize this part of talk, I believe one of the main reasons why the brain evolved a category select region is to recognize object in clusters. Normalization acting in the category select regions cause cell to respond exclusively to their preferred category in the cluster scenes and filter out non-preferred objects. Category selectivity allows the brain to decompose these complex scenes into this parallel channel for further processing. In the future, maybe one could test this computationally. Right now, the deep learning network doesn't show any category selectivities. But what if we add the recurrent connection that implement normalization? Now would the network develop the face, body, and other category selective regions? That's something we can test in the future. So category selective regions serves a very useful function purpose. Why isn't all of IT cortex organized into category selective regions? Next, I want to tell you the recent experiment I have done mapping the overall organization of IT. As I mentioned before, beside the faces, specialized network have been found in IT for bodies, scene, and color objects. But there are still a large part of IT that are unaccounted for that doesn't really show a differential response to any no stimulus comparisons. What's happening is this in apparent no man's land area? Are there additional specialized network in IT cortex beyond those already known? To address this, we began with anatomy. We electrical microstimulate a piece of this no man's land while performing simultaneous fMRI to ask what other part of brain will light up. Stimulation of the region revealed that is connected to other discrete regions in IT cortex. In total, there are 3 patches shows a significant modulation by the microstimulations. This red line shows the hemodynamic responses from the patch. They show a very clear modulation to the microstimulation. Thus, we have discovered a new network of connected patch in IT. I'm going to refer to this network as a no man's land network, even though it only fills in a part of source of IT Cortex that remains unaccounted for.
Do you see that same network when you just look at the resting state data?
It's not that clear. We do see some. Yeah, the resting state sometimes is not that as clear as microstimulation. Microstimulation do give you a much clear boundary. So resting state, microstimulation give you a clear boundary, but resting state sometimes the signal noise ratio is not high. Sometimes you can see, sometimes you do not. It's not very conclusive. Yeah. And also we even try the face patch. Even face patch sometimes it's not that clear.
You get the exact same network if you are recording from any one of those 3 sites.
That's the next slide I'm going to talk about. Sorry, can you repeat the question again? You are going to ask about E-phase.
I was going to ask if you microstimulate in any of those 3 sites, do you still get that same?
Oh, same. Yes, we do. Again, if we microstimulation this part, we also get this part and this part response. Yes, this shows they are very special connect to each other. Okay. To understand the function of this network, we did a single unit recording on the cells in these patches. At the same time, we present a stimulus set consisting of 51 objects, and each at 24 different views. And here the response of cells in this patch of the no man's land network to these 51 objects. Each row represents 1 cell, and each column 1 image. The cells are grouped separately for the 3 patches. You can see a very high consistency in the selectivity of cell within the network. All the cells are suppressed by the faces and show a very strong response to a few of objects shown here. And what do you notice about this object? We notice all of these objects seem to contain the protrusion, skinny protrusions. I'll get back to this point later. Does this network exist in every animals? Since we discovered this network by stimulating a random location outside of known networks in the first animal, we could not simply microstimulation a random site in second animal and hope to find the same network. Indeed, in next, we performed the fMRI experiment in a new monkey where we present these most and least preferred objects determined from the cell response in 3 patches. In the 3 patch in our first monkeys. And in second animal, we again saw a network consisting of multiple patch in approximately similar location as the first animals. Here the response of cell from the 3 patch in the second animal. You can see they are highly consistent with each other and with those in the first animals. So we have identified a new object network in IT Cortex that exists across animals. You might be wondering what's the difference about these 3 patches. We found a striking difference between the 3 patches in terms of viewing variance. This is illustrated by the 2 examples here. The cell in the most posterior patch was view specific, and the cell in the most anterior patch responds to the airplane across all the views. To study this view invariant object representation at the population level, here we calculate representation similarity matrix. This is 88 multiplied by 88 matrix, including 11 preferred objects and 8 views for each object. Each entry represents the correlation between the population response vector to a pair of different images. In this matrix, this diagonal line have very special meanings. They represent the correlation between the responses to identical objects, but across different views. Now, here's the representation similarity matrix from the 3 patch of the no man's land network. This dark diagonal line in the most anterior patch demonstrates that it harbors a view invariant code of object identity. This is consistent with what we found previously for the face patch. And what is network actually doing? Here again, this is the top preferred and the bottom preferred stimuli for this network. As I mentioned before, we noticed all the preferred stimuli seem to contain the skinny protrusions. In other words, could this network prefer a skinny or high aspect ratio object? To test this idea, we quantify the aspect ratio of each object as a ratio of the contour length to the area of the object image. Aspect ratio will be 1 when the image was just a circle. And when we plot the average response against the log aspect ratio of image, we found all the 3 images showing the significant correlation, suggesting this neuron do show the preference to the object of high aspect ratio. To further test this, we create a simplified stimuli set consisting of line segments, changing in the curvature, aspect ratio, and orientations. We record the neuron from NML2 and NML3, no man's land 2 and no man's land 3. These 4 groups correspond to the 4 different aspect ratio level. The neuron, do you see neuron show a very strong and a consistent preference to the high aspect ratio. They all like these skinny lines. They don't like this kind of stubby object, stubby shape. Response to this simplified stimuli set reveal a very dramatic preference to high aspect ratio segments in both areas. So one feature in this no man's land work care about strongly is the aspect ratio. So is there some way to understand what is no man's land world coding that doesn't involve looking at the preferred objects and trying to guess? Can we approach that in a more principled ways? So the key challenge here is to develop a way to parameterize objects. In other words, to describe an arbitrary with a small set of numbers. For the special cases of faces, our group was able to parameterize the faces using a pre-existing model from the computer vision called the shape appearance model that represents the face by controlling their skeleton shape and their textures. And our group found that a single cell show a ramp shape tuning to the subsets of these face parameters. And how can we do this for arbitrary objects? Until a few years ago, this still was an impossible problem. But now, with deep learning network training on object classification, we have a great solution. Deep network trained on object classification provide a very powerful way to parameterize arbitrary objects, in the sense that for every object, you can get out a few thousand number describing the object at a high level. So this 4096 number in Alexnet FC6 layer can be thought as a spanning of 4096 dimensional object space. And we can use PCA to further reduce the dimensionality of the object space. And what do the resulting object PC look like? And here you can see object with highest and the lowest projection on the first PC. It seems to correspond to the things with protrusion versus those without. And here's the second PC. It seems to correspond roughly to the animate versus inanimate. And there are many more PCs that are hard to describe in words. So now we have a sub-object axis spanning the object space. When we analysis response of cell in the no man's land network to object parameterized by this object axis, we found they behave very much like face cells, with single cell showing ramp shape tuning to subsets of object parameters. To summarize the organization and the code of no man's land network, is strikingly similar to that of the face patch network. It consists of network of connected patches. Cells within the network show a consistent pattern of selectivity. There's an increasing view invariance along the network. Finally, single cell in the network show the ramp shape tuning to the object parameters. So there seems to be a clear structural parallel between the face patch network and the no man's land network. But the no man's land network is just one network in IT. There's still a lot of IT cortex unaccounted for. As you go see here, the no man's land occupies just a small portion of the no man's land area. Are there other similar organized network in IT cortex? And if so, is there a general principle governing how this network arranged? Recall the first two axes of object space are roughly stubby versus spiky, and animate versus inanimate. Faces are stubby animate objects. Bodies are spiky animate objects. And the no man's land network seem to prefer spiky objects regardless of animacy. Recall here the top 5 preferred and the top list of preferred object of the no man's land network. This observation made a wonder. It is possible that of IT might be topographically organized according to this first dimension of the object space, in the same way that retinal topic is organized according to the polar angle and the eccentricity. And the first step to test this, we project all the stimuli that we show to the monkey onto the first 2 dimensions of the object space. Then we mark the top 100 image for the no man's land network, the body network, and the face network. You can see they approximately span 3 quadrants of the space, but there's a missing quadrant that is not represented by any of the cells. If it is true that IT is organized according to a general scheme shown here using the first 2 dimensions of the object space, then we predict there should be a force network representing object projecting strongly onto this quadrant, namely stubby inanimate objects like protrusions, lacking protrusions, for example, USB sticker or radio. Now, this is not entirely a non-trivial prediction. We have never been recorded from a single cell preferring such objects, let alone whole network of all kinds of such cells. To test this prediction, we first run an fMRI experiment with 4 different blocks corresponding to the 4 quadrants of the object space spanned by these top 2 dimensions. When we compare stubby versus other blocks, we found a network containing multiple patches. And here's the electrode entering this anterior stubby patch. And here are the responses of the 2 cells recorded from this patch. And here there's a 51 object and 24 views. Basically, you can see this vertical line showing that the response to this object across all the views. But anyway, you can see both cells prefer the stubby object, like the truck and all these houses and this basket. They all prefer the stubby objects. And here is a population plot with 5 most preferred and 5 least preferred objects shown below. You can see there's a very different pattern from no man's land network and the body patch network. So the hypothesis that IT is organized according to the first 2 dimensions of object space has actually let us discover a new network. When we project the preferred objects of the stubby patch onto the first 2 dimensions of object space, indeed, they most lie in these previously unpresented quadrants. Here you can see the 4 networks: face, body, and the no man's land, stubby and no man's land overlaid on the chrono slice in posterior IT. You can see a progression from body, face, stubby, and the no man's land in both hemispheres. And this is also true in the middle and the anterior nodes of these networks. Body, face, stubby, no man's land. Body, face, stubby, no man's land. And the organization holds across the animals. To summarize, here's a current big picture of the spatial organization of IT cortex for representing the object shape. IT consists at least 3 distinct, increasingly invariant subdivisions from posterior to anterior, with each subdivision further split into at least four quadrants the object PC1, PC space, and the sequence goes from body to face to stubby to the no man's land. In this picture, faces are not special. Previously, some people have thought face patch system arose due to the social importance of the faces. In this new picture, we believe that the face patch system arises naturally from the statistical structure of the object space. And importantly, a large swath of IT cortex follow the same principle with the similar modularities. I want to emphasize, I'm not saying each region is just coding this two-dimensional of object space. If we plot the number of cells tuned to the each object dimension, for each of this network, many cells tuned to higher dimensions beyond the first two. So the IT cortex is functionally organized according to the first dimension, but the cells are additionally tuned to many more dimensions. How rich is object code harbored by these four quadrants? To address this, we decided to reconstruct arbitrary objects from the responses of IT cell to see how good of a job could we do just cell from these four networks. Recall that I told you before that object patch show a ramp-shaped tuning to subsets of object parameters. This turns out to be incredibly convenient for reconstructing object. Because if a cell shows a ramp-shaped tuning to a subset of object features, this suggests that the response of cell can be roughly approximated by a linear combination of the features, where the coefficient of linear combination corresponds to a slope of these ramp-shaped tuning functions. Then, for population cells, we can decompose these correlation responses into response vector into a product of matrix with object feature vectors. Then if we can do the matrix inversion to predict the object features from the population response vector by a simple linear transfer function. In other words, the object feature should be equal to a weight matrix times the response of IT cells. We use the measured response to a subset of object to learn this weight matrix. Then, given a new object, we compute the corresponding object feature vector using the response object cell and learn the weight matrix. Finally, to find the object corresponding to this feature vector, we run through a new database of tens of thousands image objects that doesn't include any object we shown to the monkey. And then found the one that has a closed object feature value, and we call this object as our reconstructed objects. Here you can see some example of reconstruction using just 400 IT cells, the cells coming from the no man's land and plus a body patch, since we have the most cell from these two networks. You can say often get the size and the symmetry wrong, but otherwise they're pretty good. And here are some more examples where the form is right, but the semantic meaning is wrong. And I quite found this is quite funny. For example, the irons and the flip-flop and the helicopter and the lizard. This confirms the object feature extracted by a deep learning network is really shape-based, not semantic-based. This gives you a general sense for the richness of how the visual code contains just 400 ID cells with two specialized networks. To summarize this part of my work, cells are anatomically classed into the four networks according to the first two components of their preferred axis, forming a map of object space. This map is repeated across stage of increasing view invariance. Cells in this map harbor the sufficient coding capacity to reconstruct arbitrary general objects. But if you combine all these four networks with other known networks, we can only explain like 50% of IT. So there's clearly a lot of work to be done. But what I found is especially satisfying about this result is that they show the category selected regions are part of a continuum of specialized network. I believe they are not just representing simple categories. They are representing the regions of object space. Right now, we have a bat with a Nancy Cavendish. These things, the body patch, are just representing the bodies. We think the body patch are representing a quadrant of object space. And here are the three example objects that are not animal, but project strongly onto these body quadrants. And the dots, here the dots shows a projection of the image to the PC1, PC2 object space. And the color indicates the mean response of the cells in the body patch. And they elicit strong response in the body neurons. We are doing now human fMRI and as well as a monkey fMRI, comparing this type of stimuli to actual bodies to see which one can they evoke the strong response in the body area. So putting these two parts of my talk together, all of IT is organized into the parallel specialized network. And this allows IT to decompose the things into parallel channel to facilitate the recognition in the clusters. Some of these channels pick out a specific category, but others pick out a specific quadrant of the object space that are not confined to any of one categories. In the future, several projects along this line might be interesting to try. First one is we have only understand 50% of IT, figuring out the organizer remaining IT and provide a big picture to understand the organization of IT would be very informative to understand the Primate's visual system. Second, I want to take a step back to V4 and study information transformation between the V4 and IT. I think V4 also contains a similar object map as IT. Hopefully with a better imaging technique, such as 70 scanner, we might be see the column structures for the column structure architecture for quadrants of object space in V4. At the last, I would thank you my PI Doris Cao and thank you my collaborator Liang Shi and Mason. Also, I thank my colleagues in lab and thank you for coming to my talk. Okay. Well, what? Sorry, snake. I haven't tested the image of snakes, but it looks like some people really trying these days using a rope and a snake. They compare this one. They have a different, similar shape, but they have different semantic meaning on fMRI, on human fMRI. Right. Uh-huh.
It should be a herd of snakes. Right?
Yeah, right.
So if it doesn't care about the semantic meaning,
yeah,
then it will respond to the
Right. Exactly. Yeah, I think they will be different. I believe there will be different responses. You will see very different responses. But I haven't tested. Yeah, that's a good idea. Okay. Yeah.
If these regions are close to each other, the resolution of human fMRI only face selected regions have been found.
Yes.
Why do you think there's that? Why do you think only face selected regions have been found? It's like it's an average of all that region.
It's not region. I want to show you one thing. Sorry. There's no. It's human are very close. In human fMRI, they are very close to get something like this. Okay. There's one study from the. I'm not sure. Are you familiar with it? Natalia Kanka has found this big organization. They compare the big and the small object, and they found the whole humans. This also you can find this big area. No bigger. They basically have a gradient from the bigger object representing big and small representation difference. But if you also project all these big and small in the PC1, PC2 space, like we talked before, you can see a clear difference between this big and small in PC1. So that suggests that they also have the similar gradients, similar organization in human. And this is for his other data is 2013, 2018. All this data showing that you can see a clear difference between this, also in this PC1.
So you think the big versus small is a spurious semantic distinction, and what's really happening is just
it's just in one of the object axes, right? So I believe that's
Shows how easy it is.
Yeah. It should be, it's a more parsimonious explanation for this object.
I'm curious to have you elaborate just a little bit more on the last point you made in your discussion, this issue of columnar organization.
Yes.
Before. What's the question and what's driving that? And then based on what we do know about that potential organization, what kind of technical limits would we, what kind of technology would we need in order to see that organization?
I think, we need, first we need to increase the spatial resolution. We need to get a very clear signal for 0.5 millimeter voxel size, something like that. And we also want to reduce the point spread function for the both response. And not both, maybe both, maybe Maya, it depends on this monkey or human studies. If we get
strong motivation for why we think that interesting features would show up at the column level.
Is strong. I think the column level is something. Is some representation between the patches and the single cell. They must have certain organization to group those similar tuning features together. And the column structure is, I think, exists everywhere. You can see a column structure in almost in all the brain areas. I think that must be a very important computational unit for the brain. So, because if you saw this very clearly map structure in the IT, it cannot just suddenly appear. They must build up in these ventral visual areas. Then if go back one step back to IT's before, then I think before must have certain representation. And I hypothesis that might be is have the column structure in before. So that's something we need. I think it's worthwhile, worth trying to see. Yes.
So here you train the deep neural network and then you use code and then use and then you compute the correlation and then assumption and so on.
Yes.
Then what if you train another network and then you use some other techniques to reduce the dimension, and then probably other neurons will be correlated with the new representation. So every time.
I see.
How do you deal with this?
Okay. I see. I want to show you some result we compute. So here's 8 different network we tried. We tried this is the name is on the bottom. You can see there's 8 networks, and if you present all these 4 same map like we presented before for these 8 different networks, you clearly see there's still clearly see these 4 progression from the this is stubby, there's no man's land, then this is body, this is face. They still group together, no matter which network you use. They have all kinds of different structures. That means this representation, they do have some intrinsic statistical structure, which is not depending on which kind of network you use to train. Yeah.
So you took you got these maps.
Yes.
2-dimensional space. Taking the first 2, but of course, presumably there's a third.
Yes.
Principal component which has significant power.
Right.
And you would then distribute across the 3-dimensional space.
Right.
Do we have a strong set of intuitions for why we really think this kind of 2-dimensional space is fundamental?
Right.
So just a convenient way that we like to look at the world. So we stop it.
That's also a very good question. I tested with, so I don't know if I have the figure here. Maybe I don't. The question is, we tested this with our data. We first tested with using this k-mean analysis to see how many clusters can we see in all the PC space. Then we found that there's 4 clusters. But if you remove the PC1, the PC2, and can we see more cluster in the remaining data structure? If we remove the PC1 and PC2, we don't see any further cluster in other PC dimensions. So based on our current data, we can say now the neurons are clustered into the PC1, PC2. But in the future, maybe with a more fine structure, with more recording, and maybe we can find more. I don't know. So far, the data doesn't support that we have more. We only see the cluster in the PC1 and PC2 space. We don't see in the other spaces. Yes.
So I wanted to revise my previous question. Is that nonlinearity, and I guess I really mean recurrence. So your feed forward, I don't think you can get, as Roger says, what you have the ability to winner take most. I don't think you can get winner take all without recurrent connectivity.
Without the recurrent current.
Yeah, so I'm wondering whether, I mean, are you asserting that the flow of information is purely feed forward?
No. It's the normalization indeed. Maybe I did not explain very clear. The normalization itself is a lateral inhibition model. It's a lateral inhibition model. It's not a feed forward model. It's all these things. All these things is showing this is the input. Okay.
So I think the prediction would be, again, just like personal communication logic to tell.
Okay.
Like 8 seconds ago.
Okay.
Would be that you see some response to the non-preferred category. It's not really winner take all, it's winner take most.
To the most, I think.
Would you see evidence of that?
We don't see that for the winner. We don't see it. So in our data, it's a very little, very subtle responses for the object. But here I want to emphasize this inhibition here is a lateral inhibition. It's a recurrent network action. It's not a pure feed forward. But you can also have feed forward inhibition, but it also can recurrent network will also provide inhibition.
Right. Yeah. Right. So to get like pure winner take all, either 1 for something and 0 for something else. You either need like a threshold nonlinearity, which is non-continuous, or you need kind of several cycles in recurrence in order to suppress the like ceramic.
Okay, yeah, the pure. That one we haven't thought about this. Why is a pure, right? What do you mean by pure winner take all? Yeah, it's just like roughly we can say we can get a winner take all. Yeah, that's yeah. Okay.
In your diagram, 4 different patterns on the brain. You know, I know you're just trying to do a diagram, but you're sort of blind.
Let me. Okay, hold on. Let me open that.
Anyway, I mean, only thing about that kind of map is that the ones on the outside can't touch the diagram.
The diagram. Okay, yeah.
I'm just saying, you know, maybe it's a.
I see. Right.
The 2 outermost can talk to each other.
Okay. Yeah. That's also one possible. Yeah, that's probably I want to go back to look into more carefully about that. That's also possible, right? Yeah, right. Yeah.
And sort of on that topic, would you expect you'd see actual anatomical connectivity using dye in histology?
Yeah,
between the.
Yeah, I would. Exactly. I will. The people have our lab has other people has done this on the face patch. They see the very specific connection within this face patch using all these anatomical tracer techniques.
So you would expect to see.
I will expect to see exactly. Yeah.
Is it within the power of diffusion MRI to see this?
That one I don't know. I don't know diffusion. I'm not sure if diffusion MRI.
What evidence is there in humans for this same level of organization? Has that just not been done yet?
You're right. I show you that before. This big and small is one evidence to showing that because they also see this large object versus small object can be very explained by this PC1 space, and they also found the orthogonal one is animate versus inanimate. The big and small and the animate and inanimate kind of is orthogonal diagonals. I think those are very strong evidence to support our model, and also we are also trying to applying this. We are working on this, applying this model on the human fMRI to see if we. So far, we already can clearly see based on our same analysis. We also see the face part, those face region and the body region and the scene region is fitting our story very well. Yeah.
So 2 quick questions. One is, what about if you're going to translate this to the human and the role of development, for example?
Yes.
How much has these monkeys actually been behaviorally trained to a certain criterion when you look at the distinction between face and objects, for example? And then if they have a lot more experience, the age of the monkey, and similarly therefore the
structure. Yeah, that's a good question. I think this development question, Margaret Livingstone, they did something like that. They, for example, they can shut down the face input. They don't let monkey to see the face at all. Then they don't see the face patches. So I also think this the map we saw here is based on the visual experience. That means the monkey needed to extract those static structure based on their previous visual features. Based on their previous, I have to see this object, then I build a PCA on this object space, then I can get this area. If you don't see some, for example, let's say if we shut down some stimuli, if we don't let the monkey to see the body, to see the animal, don't see the faces, then this whole map will change a lot. Our monkey is born naturally, was also raised naturally. They saw every kinds of things. So I don't know if we, what?
Would you imagine in Marge's kind of face-free monkeys? In fact, they would also have deficits of the ability to perceive or process other parts in the PC1, PC2.
Yeah, I would, I would expect the same thing. I would expect, right. That sounds like.
On that note, I just ask also the functional significance of what they see. So they may have exposure to something.
Yes.
But it doesn't have meaning for it. So say, for example, the face of a human being, until a monkey gets enough exposure and interaction with it, may not have the same statistical characteristics.
I see. That's why I don't know. I'm not sure. I think if you don't have a meaningful object, lots of objects for our monkey also doesn't have meaning. They never seen some certain object before. But the neuron still show response to those objects. It looks like the neuron, at least at neuronal level, it looks like those neurons responding to the shape more than the semantic. That's what I can say now. Yeah, I don't know.
Okay, if there's no more questions, let's thank Pinglei again.